Okay, so there's two different terms. One of them is called monomer, and the other term is called polymer. And as usual, in order to memorize all those terminologies, which I know that they are a lot, you have to split the word. What do you mean by that? Mono means one. Poly means many. But not just many. Many repeated. So as if you are saying that a monomer, it's a unit. One unit in math is for word unit. Unit. So, one unit. Polymer, one, many, repeated units. So, if the building blocks here are called the nucleotides, and you just would like to form just one building unit, so the building unit, another name for it, or a description for it, it will be a monomer. A monomer. One unit, one building unit. Like nucleotide plus nucleotide plus nucleotide, this would form the polymer. Many repeated units. Nucleotide plus another nucleotide plus another nucleotide. One monomer plus another monomer plus another monomer. Those are repeated units. So, it will form the polymer. Nucleotide is a building unit, and the polymer of the nucleotide is the DNA itself. Is the DNA itself. So, DNA is the polymer, its monomer is nucleotide. حلوة. This nucleotide, this building block, is formed of some subunits. Okay, yes, it's the building unit, but it's formed of other 3 units working together to make this one entity. So, nucleotide is formed of 3 subunits. Those 3 subunits look like this. 1, 2, 3, 4, 5, and this is the sugar. 5 carbon sugar. 5 carbon sugar. At one side, the 3' side is attached to a nitrogenous base. And the other side, the 5' side, is attached to a phosphate group. Phosphate group. So once again, to recap, كده a nitrogenous base, link it to a sugar, link it to a phosphate, they will entirely form a nucleotide. Nucleotide is a building unit of DNA. So a nucleotide plus another nucleotide plus another nucleotide, they will eventually form the polymer, which is the DNA. The kind of sugar in the DNA is called deoxyribose sugar. So here, the kind of sugar is called deoxyribose sugar. That's where actually the name of the DNA is coming from. It's called deoxyribo. Deoxyribo, as if I'm saying that, the kind of sugar in DNA is deoxyribose sugar. Now, what about the bases? Those bases, their arrangement will give you the specific protein you are aiming to. Those kind of bases, whether subdivided into. Based on their structure, whether to be one ring, formed of one ring structure, or they will be formed of 2 ring structure. If they are just formed of 1 ring structure, they will be called pyrimidines. And if this base is formed of two ring structure, is structure خلقت ربنا خلقتها كده. Two rings كده joined together. Two rings being joined together. Here it's only one ring. It's only one ring forming the structure. They will be called pyrimids. The pyrimidine bases are A and G. And the purine bases, no, sorry. The pyrimidines are C and T. And the purine bases are A and G. Let's give kida what each one of them standing for. C is called cytosine. T, thymine. A, adenine. G, guanine. And in order to memorize it, which is pyrimidine and which is purine, cytosine contains Y, thymine contains Y, and pyrimidine contains Y. So C and T will be subgrouped as pyrimidines. Like, how can we complement each other? How can the DNA complement? Because DNA is formed of a double helix. It's two structures. Not only one structure, it's two structures twisted around each other. What makes them really twisted around each other? The bases, whether A and T, C and G will be complementary to each other. So every A will be complementary to T and every C will be complementary to G. Every A will be complementary to T. So what's this kind of bond? Joining the two bases together, hydrogen bond. Hydrogen bond. What about this sugar and this phosphate? What do you call this kind of sugar, bond? Covalent bond. Covalent bond. طيب هنا كده I'll make some sort of comparison for you. So in the DNA structure, you have two kind of bonds. Number one between the bases, and this is called hydrogen bond. Number two, between deoxyribose sugar and the phosphate group. And this kind of bond is called covalent bond. Covalent bond is a very strong bond. Strong bond is unbreakable. You have a sugar phosphate backbone and it's a backbone. But what about hydrogen bond? It's breakable, it's weak. It's a weak bond. So, if this is the DNA, one strand and another strand, and then you are having here, let's draw it as if you are drawing in a form of nucleotides. Phosphate, joined to sugar, phosphate, joined to sugar, like this. Okay? Like what about here? It will be sugar برضو. Phosphate, joined to sugar, phosphate, joined to sugar. This part, the external part of the DNA is called sugar. Phosphate backbone. It's the backbone of the structure. So what about the last part of the nucleotide? It's the base. Base is here. If here is A, it will be complementary to T. If here is T, it will be complementary to A. If here is C, it will be complementary to G. And the kind of bond joining the two strands together is hydrogen bond.